This is Unit 2A, Part 2. We're going to continue with renal physiology, uh, physiology of the urinary system. One of the major measurements or a key measurement of renal function is looking at the glomerular filtration rate, or GFR. GFR is the total amount of filtrate that's formed every minute by the kidneys, and there's some key factors that govern the rate of formation of that filtrate. One is the total surface area available for filtration. The other is the permeability of the membrane through which that filtration occurs. And the last one is the net filtration pressure, which we discussed in the previous video, Unit 2A, Part 1. In terms of glomerular filtration, the rate of formation of filtrate by the glomeruli is directly proportional to the net filtration pressure. So changes in GFR normally occur with changes in glomerular blood pressure. Glomerular blood pressure, of course, is a major factor in determining what NFP, net filtration pressure, is. If glomerular filtration rate is too high, then it's possible for needed required molecules that get into the filtrate to not be reabsorbed quickly enough, and they therefore get washed out or lost in the urine. Remember that a lot more filtrate is formed than actually gets out as urine. Much of what goes into the initial filtrate being formed will be reabsorbed back, but if filtrate is being formed too quickly, then the flow rate through the tubular system of all the glomeruli together will be too quick, and there's not enough time then for reabsorption to occur, and those molecules will get lost in the urine. If, on the other hand, glomerular filtration rate is too low, then the fluid that goes into the filtrate will spend quite a long time moving through the tubules, and too much gets reabsorbed, including even some metabolic waste products. And so there is a sweet zone for the appropriate level of glomerular filtration rate. Neither too high nor too low is ideal. And that's how the body tries to regulate GFR. Three mechanisms that are key to controlling GFR are called renal autoregulation, which is an intrinsic mechanism, or it's intrinsic to the kidneys, built into the kidneys. Also neural control mechanisms, and the last one is hormonal mechanisms. And the hormonal system is the renin-angiotensin system, which we've discussed a little bit, but we'll talk about in more detail. Now, these kind of mechanisms, regulatory mechanisms, are not simple, and there's quite a bit of detail here. We'll go through it. The first, or renal autoregulation, as I said, is a built-in intrinsic system. Under normal conditions, autoregulation by the kidneys on their own function is enough to maintain a nearly constant appropriate GFR. Autoregulation involves controlling essentially the diameter of the afferent and the efferent arterioles that go into and come out of each of the glomeruli in the two kidneys. You have two mechanisms, or two types of control that are autoregulatory. One is the myogenic reflex, and the other is called flow-dependent tubuloglomerular feedback mechanism. A mouthful, but we'll go through each one. These are the two mechanisms involved in autoregulatory control of GFR. The myogenic reflex is a response to changes in pressure in renal blood vessels in the inflow of blood into the kidneys, particularly into the the glomeruli. And it occurs as a result of the inbuilt response of smooth muscle to being stretched. Smooth muscle will contract when it is stretched, and if the pressure, the blood pressure in a vessel is higher than normal, it will tend to stretch that blood vessel out, and the blood vessels have smooth muscle around them, and they will contract automatically in response to that stretching. So if the overall systemic blood pressure rises, it will cause afferent arterioles that feed blood into each of the glomeruli to constrict, and you'll get vasoconstriction, thus reducing blood flow to the glomeruli. This prevents glomerular blood pressure from reaching potentially damaging high levels. Remember, if the blood pressure in the glomeruli is too high, if afferent arterioles are too wide and feeding in blood, the blood flow is easily going into them, and the pressure inside the glomerulus is too high, then you'll get a abnormally high rate of filtration, glomerular filtration rate, and you'll start losing important molecules out into the urine. So this is one way that this myogenic reflex is one way that controls that. If the pressure is too high, you get afferent arterioles constricting. If the systemic blood pressure is too low, then that will trigger afferent arterioles to dilate and allow more blood to flow in, and therefore allow for a higher glomerular hydrostatic pressure, and therefore a higher net filtration pressure. And so it brings it back into what I called the sweet zone. So here, the second control mechanism that mediates autoregulatory effects is flow-dependent tubular-glomerular feedback. This mechanism senses changes that are occurring to the juxtaglomerular apparatus. I remind you that the juxtaglomerular apparatus, the key cells involved in it are the juxtaglomerular cells around the afferent arteriole as it enters into the glomerulus, and also there's a few juxtaglomerular cells apparently around the efferent arteriole. These cells, in combination with macula densa cells that are in the wall of the distal convoluted tubule, where it comes close to the glomerular capsule that the tubules originated from, these together form the juxtaglomerular apparatus. Now, what's happening here is that macula densa cells are always sensing the nature of the fluid that's in the distal convoluted tubule. When the macula densa cells detect a slow filtrate flow or filtrate with a low osmolarity, then they will induce vasodilation of the afferent arterioles. So if the flow of filtrate in the distal convoluted tubule is too slow, that means that the GFR is too slow, that filtrate formation is too low, and the fluid flowing through the tubules is too much of it, and too many molecules are getting reabsorbed, so it has a low osmolarity. Then the macula densa cells are able to induce vasodilation of the afferent arterioles, which would increase blood flow into the glomerulus, increase therefore the hydrostatic pressure, glomerular hydrostatic pressure, and therefore increase net filtration pressure, and therefore increase glomerular filtration rate, increase blood flow, increase in GFR, allowing more flow, more glomerular filtration rate rises, so more filtrate is being formed and more fluid is going through the tubular system. There's less time for filtrate processing. And remember, filtrate processing means essentially reabsorption. If the opposite situation occurs and the macula densa cells that are monitoring that flow rate in the distal convoluted tubule, if they detect a rapid filtrate flow or filtrate with a high sodium chloride, the filtrate is forming at too high a rate and it's flowing through and not enough sodium or chloride are being reabsorbed, then it'll be high filtrate flow and high electrolytes, then the macula densa cells can induce the juxtaglomerular cells, which are also part of that juxtaglomerular apparatus, to secrete a chemical compound which has vasoactive, and it has vasoconstrictory effects on the afferent arterioles. So with GFR, slow filtration flow, you have too low a GFR, the macula densa cells can trigger a vasodilation of the afferent arteriole, causing more flow of blood in. Or if the GFR is too high and there's lots of filtrate flow, then the macula densa cells can cause the juxtaglomerular cells to release a compound that causes vasoconstriction of the afferent arterioles. So the end result is either vasodilation of the afferent arterioles to increase GFR, or vasodilation of the afferent arterioles to, vasodilation of the afferent arterioles to increase GFR, or vasoconstriction of the afferent arterioles to decrease GFR. Thus, with the vasoconstriction, reducing flow, decreasing GFR, allowing for more filtrate processing. And so in the end, what you have is a mechanism, this flow-dependent tubular glomerular feedback mechanism, controls the diameter of the afferent arterioles, either causing them to widen, so flow increases, or to constrict, so that flow decreases, once again keeping them in the sweet zone. So this is a mechanism that is intrinsic to the kidneys, built into the kidneys. This mechanism is one of the two. The other one is the myogenic reflex, which simply is the smooth muscle around the afferent arteriole. And this one involves chemical mediators, molecules that will induce either vasodilation or vasoconstriction. Those are the intrinsic mechanisms that are built in autoregulatory effects. Under normal conditions, autoregulation can maintain a near constant GFR in that sweet zone of values. And they'll do this by maintaining a near constant blood flow through the kidney glomeruli, even if the systemic arterial pressure, in other words, the renal artery, the left and right renal arteries that feed arterial blood into the kidneys, even if that pressure is ranging as low as 80, systolic as low as 80 or as high as 180, these mechanisms will regulate and narrow the pressure inside the glomerular afferent arteriole so that the glomerular pressure is appropriate for appropriate function and a near constant GFR. So it's an autoregulatory effect that can, even when the systemic blood pressure varies. Now, those were intrinsic controls. The other two types of controls are extrinsic controls that will maintain, regulate GFR. One being the nervous system controlling it, and the other being the renin-angiotensin system. We'll go through these extrinsic controls one at a time. The sympathetic nervous system has an effect in that with sympathetic nervous system at rest, in other words, when a person is relaxed and under more parasympathetic, has more parasympathetic output from their nervous system. Renal blood vessels are typically maximally dilated, and autoregulatory mechanisms are the dominant force that are regulating the GFR rate and maintaining it within that appropriate zone, neither too high nor too low. However, under stress, with an increase in sympathetic output, norepinephrine, the neurotransmitter norepinephrine, is released by the sympathetic nervous system, and epinephrine is released by the adrenal glands, specifically the adrenal medulla. And so these two neurotransmitters, sympathetic neurotransmitters, can have an effect on the afferent arterioles to cause them to constrict and therefore decrease flow into the glomeruli, thus decreasing the glomerular hydrostatic pressure, the blood in the glomeruli. Remember, that's the capillaries within the Bowman capsules, or the glomerular capsule. So it decreases afferent arterial constriction. Vasoconstriction will decrease hydrostatic pressure in the glomerulus, decreasing net filtration pressure, therefore decreasing GFR, glomerular filtration rate, the rate of formation of filtrate. So filtrate formation gets inhibited. I'm going to talk, the next thing I'll talk about is for extrinsic controls is the renin-angiotensin mechanism or system, but I should tell you that the sympathetic nervous system can also stimulate that on its own as an extrinsic trigger. But the renin-angiotensin mechanism on its own also has an effect. Renin is a hormone that is synthesized by juxtaglomerular cells, those cells that form part of the juxtaglomerular apparatus and are around the afferent and efferent arterioles, just where they go into and come out of the glomerulus. The release of this hormone, renin, is triggered by reduced stretching of the juxtaglomerular cells. They are granular in that they contain granules of hormones, of the hormone renin in them. So you have a reduced stretching of those cells because the blood, and that typically happens when blood pressure is low in the afferent arteriole. So the afferent arteriole isn't stretched out as much, and therefore the juxtaglomerular cells around it are not stretched as much. Renin release is also stimulated by activation signals from the macula densa cells that are in the wall of the distal convoluted tubule near the glomerulus. Also, and this was what I mentioned earlier about sympathetic stimulation, the sympathetic nervous system can directly stimulate renin-angiotensin, renin release from juxtaglomerular cells. So this is a sympathetic adrenergic. It's epinephrine kind of response. So these are 4 ways that renin release can be triggered, and the response to that renin, and remember the renin is released from the juxtaglomerular cells. The response to, did I say 4? I meant 3, sorry. What happens is when you have release of renin from juxtaglomerular cells, renin will act on a plasma protein, which is inactive, as indicated by its name. If a protein molecule ends in OGEN, then it's indicating that that protein is inactive. So renin will act on angiotensinogen, a plasma protein, and activate it to form angiotensin 1. When it says to release axon angiotensinogen, to release angiotensin 1, what's really happening is that the renin acts as an enzyme and cuts or cleaves. Cleaves or cuts meaning the same thing. It cuts part of the molecule off, releasing this part of it, which has activity. this now has enzyme, this protein, angiotensin 1, has activity. Angiotensin 1 then floats through the plasma, and when it gets to the lungs, it gets converted to angiotensin 2, again by a enzyme that cuts the molecule. The enzyme in the lungs that cuts the molecule, this so-called conversion, it's really a enzymatic cleavage or cutting of the protein angiotensin 1 to release angiotensin 2. That's mediated by an enzyme called angiotensin converting enzyme, ACE, angiotensin converting enzyme. All right? Now, angiotensin 2, once formed, has a number of effects. It will cause the mean arterial pressure to rise because it triggers, it has quite a strong systemic, that means throughout the systemic circulation, it has a fairly strong systemic vasoconstrictory effect. It will cause vasoconstriction. If the systemic circulation constricts overall, then there's less space for the same amount of blood and the pressure rises. So this is a way of raising blood pressure. It also has an effect on the pituitary gland. It will trigger the release of antidiuretic hormone from the posterior lobe of the pituitary. As you know from last semester, ADH, or antidiuretic hormone, is stored and released is stored in and can be released from the posterior lobe of the pituitary gland in the brain. Antidiuretic hormone has an effect to increase uptake or reabsorption of water, again helping with maintaining blood pressure and even raising blood pressure. The other thing that angiotensin 2 does is that it will stimulate the adrenal cortex to release a hormone called aldosterone. Now, what aldosterone does is it causes increased reabsorption of sodium in the kidneys. And when you have an increase in sodium reabsorption, you also have, along with it, following it, hand in hand, increased water reabsorption. And that's what I mean by water follows. Increased sodium reabsorption will be accompanied by increased reabsorption of water, again helping to maintain or raise the blood pressure. So as a result of renin acting on angiotensinogen, you get formation of angiotensin 1 here, and angiotensin 1 being acted on by ACE. When it gets to the lungs, ACE is present there, and ACE will convert it to angiotensin 2, and angiotensin 2 does each of these things. It causes systemic vasoconstriction, raising blood pressure, stimulates release of antidiuretic hormone, helping to raise, again, blood pressure, and stimulates adrenal cortex to release aldosterone, which triggers increased reabsorption of sodium and water, again, raising blood pressure. So both the systemic hydrostatic pressure and the glomerular hydrostatic pressure will rise, and therefore the net filtration pressure rises, and therefore the GFR rises, and what you have is GFR returning to the normal range. Okay? Complicated. Under tight, careful control, both intrinsic control and extrinsic control. We've covered mechanisms of intrinsic control, and here we cover two mechanisms of extrinsic control. And all that I've said here, or a lot of what I've said here, is summarized in this table, and you should use this table as a way of reviewing what I just talked about in terms of regulation of GFR. This, what's described here in this flow chart, is regulation of the glomerular filtration rate when blood pressure is too low. And I remind you that blood pressure will be, if blood pressure is too low in the renal blood vessels, the afferent arterioles, then again the net filtration pressure will be too low, and therefore the GFR will be too low, and therefore the formation of filtrate will be too low, and so the fluid in the filtrate is going through the tubular system, and too much of it's getting reabsorbed. So there has to be a correction, regulation of that. And these are the mechanisms, intrinsic and extrinsic mechanisms that regulate GFR. And I've gone through each of these, and you should, follow them through. Remember, the end result is it started with too low blood pressure, therefore too low GFR, and the end result needs to be an increase in GFR to bring it back into the normal levels. And each of these pathways is operating and are mechanisms or control mechanisms that help regulate the GFR. So I would use this as a, go through it and use it as a review for that, and it should all make sense to you. I've mentioned all of these things in the previous slides. Okay, here we are. Let's move on. Are there other factors that affect glomerular filtration? Yes, and a few of them are listed here, and we're not going to get into much detail. There are some certain prostaglandin molecules that can affect GFR. They have a vasodilatory effect. They can act as vasodilators, and they can be produced in response to sympathetic stimulation and also in response to angiotensin II. They can help prevent renal damage if the peripheral resistance is increased. When peripheral resistance increases, the resistance of the blood vessels, the pressure gets too high, and you can get too high a pressure, so vasodilation, systemic vasodilation is good. I don't want to get into too much detail about these, but you should be aware of them. Nitric oxide is a molecule that can cause vasodilation. It's produced in the vascular endothelium and has a localized vasodilatory effect. Adenosine has a vasoconstrictory effect, and another molecule called endothelin has a vasoconstrictory effect. These are other factors that have effects on GFR by changing, blood vessel diameter and therefore pressure. Prostaglandin, nitric oxide, adenosine, and endothelin. Okay, let's talk a little bit about reabsorption. We were talking before about, remember, there are three major processes, key processes to the formation of urine. Three key mechanisms or processes that allow the kidneys carry out. One is filtration, so formation of filtrate. The second is reabsorption, and the third is secretion. And we just talked about filtration, and we talked about regulation of filtration, so now let's talk about reabsorption. Reabsorption is a process that occurs in the tubular system. It occurs, and it's important because the filtrate that is formed, as I've told you before, is in way too big a volume. You can't lose that kind of fluid in your urine. You would quickly dehydrate and lose too many electrolytes. Most of the filtrate, in fact, the vast majority of the filtrate gets reabsorbed back into the blood from the tubular system back into the blood. And that reabsorption occurs from the tubular system into the peritubular capillary bed. That's second blood capillary bed after the first one, the glomerulus. These transported substances or reabsorbed substances will move through three membranes to get back into the bloodstream. They move across the cells that surround the tubule, and then they move through the basement membrane, and then they move through the endothelial cells that line the peritubular capillaries to get into the bloodstream, in the blood in the capillaries. I don't know if I'm going to talk about this mechanism. Let's move on. Okay, so what route do you have? There are three routes. One is between the cells. One is through the cells. I'm sorry, there are two, yeah. There are two major routes. One is between cells here and here, and this, and then there's through the cells. So crossing into the endothelium, the cell that lines the tubule, and then across the endothelial cells that line the capillaries. So on the left-hand side of this diagram, that's the space of the tubule, and we're looking, actually, I want to remind you here that we're looking at the proximal convoluted tubule. Here you get a lot of reabsorption occurring, and the key things that are reabsorbed are, of course, the key things that are reabsorbed are water and solutes like electrolytes, and it can be between the cells or through the cells, paracellular, transcellular. And you go from the space of the lumen of the tubule across the cells that line the tubule, and then across the endothelial cells that line the capillary to get into the bloodstream. So, during tubular reabsorption, what you have happening is that any organic nutrient molecule like amino acids or any simple small monosaccharide carbohydrates like glucose that manage to get into the filtrate will be reabsorbed. Normally you don't get any amino acids or carbohydrates like glucose coming out into the urine. Anything, and it should be a small amount, that gets in will get reabsorbed. At the same time, you also have water and ions, electrolytes, being reabsorbed, and that's under hormonal control. There are active and there are passive processes. Some processes require energy and others don't. Let's talk first about sodium reabsorption. Sodium reabsorption is always active, requiring energy. It's actively transported across by a sodium-potassium pump action. Okay, in terms of reabsorption that's occurring, sodium reabsorption is occurring in the tubules, and it is an active process, and I don't want to get into too much detail on this. Suffice it to say that if the capillaries have a low hydrostatic pressure, they can take up more sodium, or if they have a high osmotic pressure, then they'll take up more sodium. Low hydrostatic and high osmotic pressure. These tend to cause sodium to come across, and sodium typically moves with other solutes. I don't want to get into this too much. In fact, I don't think I want to go into too much detail. Although I do want to tell you and point out to you that when water moves across, when sodium moves across, the reabsorption of sodium, it's always accompanied by water, okay? And the water will move across by osmosis. And when water moves across, it has to go through pores, and those pores are called aquaporins. Okay, I'm not going to get into the rest of this. It's enough detail. There's really too much detail, I think, for you guys. You know, it's enough to know regulation and understand. I don't want to get into all of this. This is reabsorption in the proximal convoluted tubules. Depending upon where we are in this tubular system, there are different kinds of reabsorptions occurring. Okay. There are some molecules that don't get reabsorbed, or their ability to be reabsorbed can become saturated so that you can't reabsorb anymore, and anything past that level gets excreted out. So everything that does get reabsorbed has what's called a transport maximum, or TM. And that's because you have carrier molecules that are involved in the reabsorption. So you have a fixed number of carriers that can help with reabsorption of molecules back. There's carriers for nearly every substance that is actively reabsorbed. And if they become saturated, because there's so much of the molecule that they normally reabsorb or involved in reabsorption, then the excess gets excreted out into the urine. All right. So, as I mentioned earlier, amino acids or glucose will be reabsorbed, but if you're spilling a lot of glucose or amino acids into the filtrate being formed, and that can happen in certain pathological states, especially with very high blood pressure, extremely high blood pressure, or chronic high levels of glucose in your plasma, then so much of those substances get into the filtrate that carrier molecules can't keep up the pace that the new glucose or amino acids are arriving, and therefore can't reabsorb everything, and it starts to spill out in the urine. Anything that is normally not reabsorbed at all is because there are no carriers, or they can't cross on their own through the lipid membrane. Anything that's lipid soluble normally would go straight across. So substances that can't be reabsorbed, there's no carrier, or they're not lipid soluble, or they're just too large to go through the pores. Urea, which is a nitrogen waste product, or metabolic byproduct, waste byproduct, normally is the main nitrogen containing molecule in the urine that the body gets rid of. But it's not all. If it's in really high amounts, it can get partially reabsorbed. There is a molecule, it's important to understand this in terms of kidney function and renal physiology. There is a molecule called creatinine. It is never reabsorbed. It is not reabsorbed at all. And so it actually acts as a good marker, because you form creatinine at a steady pace. In the body, there's a steady production of creatinine, and creatinine is always excreted out in the urine, and none of it gets reabsorbed, so there should be a steady release of creatinine in the urine, and creatinine should therefore never accumulate in the blood plasma. The other thing that doesn't get reabsorbed is uric acid. We'll talk about that. We'll talk about urea and uric acid probably later, but it's important to understand this idea of creatinine, I would say. In fact, a good measure of overall renal function. is creatinine, what's called creatinine clearance. Okay, the reabsorption in the tubules and in the collecting ducts is not exactly the same. In the proximal convoluted tubule, sodium is getting reabsorbed, and any nutrients, I've mentioned that before, and there's also other cations and anions and water, and also small proteins that might have worked their way managed to get into the filtrate. In the loop of Henle, there's an important difference between reabsorption in the descending limb of the loop of Henle and the ascending limb. The descending limb can reabsorb water, but the ascending limb does not reabsorb water. And I'll show you in a slide in a few minutes what that ends up causing. It's easier on a diagram to go through these kind of reabsorption differences between the proximal convoluted tubule, the descending and ascending arms of the loop of Henle, and the distal convoluted tubule. The distal convoluted tubule does absorb electrolytes, both anions and cations, and also the collecting ducts at the end can reabsorb water and urea. I don't want to get into this detail at all. I think we should leave this alone. Okay, we're on to a different topic. Part of regulation of sodium levels in the body, and sodium is under tight control, are hormones. I remind you the hormone aldosterone that's produced in the adrenal cortex causes a increase in reabsorption of sodium and water along with it. Water follows from the filtrate in the kidneys. There is another hormone which has the opposite effect. It allows for increased release of sodium and therefore water. Okay, and that protein, or it's a protein hormone, is called atrial natriuretic peptide, or ANP. I just want to point out to you that the abbreviated symbol for, or abbreviation or symbol for sodium is Na, and Na actually stands for natrium. Natrium is another name or term that can be used for sodium. In some countries, sodium isn't called sodium, it's called natrium. So Na stands for natrium, sodium, and natriuresis is the release of sodium. Notice it's a protein, it's a small protein, so it's called a peptide. And notice here it's called atrial, and that's because it's produced in the wall of the atrium in the heart. It's a major regulatory factor in regulating extracellular fluid volume, the fluid volume in the vasculature, because it can, when released, it can trigger natriuresis, release or excretion of blood sodium into the filtrate, so loss of sodium in the urine. It does a number of different things. It can lower the blood sodium, as I said, by acting directly on the tubules to cause in the medulla, the collecting ducts in the medulla, to inhibit sodium reabsorption, so more sodium spills out in the urine. It also inhibits renin secretion. I remind you that renin secretion triggers angiotensin 1, which forms to angiotensin 2, and angiotensin 2 can trigger aldosterone release, and aldosterone causes sodium reabsorption, so it inhibits renin, inhibiting therefore sodium reabsorption. It counteracts rather, in fact, all of the effects of angiotensin 2. So it inhibits sodium reabsorption, more sodium goes out of the tubules in the form of urine, inhibits renin secretion, and therefore lowers blood sodium that way, and counteracts the effects of angiotensin 2. In this way, you have indirect stimulation of an increase in the glomerular filtration rate, and thus reduction in water reabsorption. When you raise GFR, you're going to raise the rate of formation of the filtrate, and more fluid will be passing through the tubule, and not all of it gets reabsorbed, and therefore you're increasing the loss of fluid and sodium, and therefore it acts to decrease blood volume and in that way lower blood pressure. Quite a significant protein. It was discovered, in fact, in the Department of Pathology at Queen's University by this fellow, Adolfo de Bolt, who soon after left Queen's and moved to the Heart Institute in Ottawa, became director of the Heart Institute, or the cardiovascular endocrinology lab there. A very important discovery. This protein hormone, by the way, it is not surprising that it's, in the wall of the atrium. If blood pressure is high, you want to reduce blood volume and therefore reduce blood pressure, right? When blood pressure is high, you get an increased stretch in the wall of the heart, in the atrium, the atrial wall of the heart, triggering release of this protein. So the protein has many effects to cause release of sodium and water, dropping the blood pressure. It's a nice system. Okay, that was reabsorption. Now we'll talk about secretion. Remember that filtration is a process. The three processes are filtration, reabsorption, and secretion. And filtration is the formation of the first fluid into the tubular system, moving from the blood plasma across the wall of the glomerulus into the glomerular capsule to first enter into the tubular system there. And then a lot of that ends up being reabsorbed as it moves along. But then you have a third process called secretion, which is the secretion of molecules from the bloodstream of the peritubular capillaries into the tubules, further down the tubular system. And we're going to cover tubular secretion in the next video, which will be Unit 2A, Part 3. So this here is the end of Unit 2A, Part 2.